Sundar sir. Hello.
Hello.
Sundar sir.
ಹೇಳಿ.
ಯೂಟ್ಯೂಬ್ ಅಲ್ಲಿ ಜಾಯ್ನ್ ಆಗಿದ್ದಾರೆ. ಹಾಗಾಗಿ ನಾವು ಸೆಷನ್ ಸ್ಟಾರ್ಟ್ ಮಾಡೋಣ ಸರ್. ಸರ್.
ಹೇಳಿ.
ಸರ್ ನಾವು ಸೆಷನ್ ಸ್ಟಾರ್ಟ್ ಮಾಡೋಣ ಸರ್. ಓಕೆ. ಸೊ ಇಲ್ಲಿ ಬಂದಿರುವಂತ ಎಲ್ರಿಗೂ ಸ್ವಾಗತವನ್ನು ಬಯಸ್ತಾ, ನಮ್ಮ ಸುಂದರ್ ಸರ್ ನಿನ್ನೆಲ್ಲ ಮೊನ್ನೆ ತಾನೆ ಅವರು ಇವಲ್ಯೂಷನ್ ಬಗ್ಗೆ ಟಾಕ್ ಕೊಟ್ಟಿದ್ರು. ಇವತ್ತು ಅದರ ಮುಂದುವರಿದ ಭಾಗವನ್ನ ನಮ್ಗೆ ಪ್ರೆಸೆಂಟ್ ಮಾಡ್ತಾ ಇದ್ದಾರೆ. ಅವರಿಗೆ ಸ್ವಾಗತವನ್ನು ಕೋರುತ್ತಾ, ಮತ್ತೆ ನೆರೆದ ಎಲ್ರಿಗೂ ಈ ವೆಬ್ಎಕ್ಸ್ ಅಲ್ಲಿ ನೆರೆದಂತ ನನ್ನ ಸಹಪಾಠಿಗಳಿಗೂ ಮತ್ತು ವಿದ್ಯಾರ್ಥಿಗಳಿಗೂ ಸ್ವಾಗತವನ್ನು ಕೋರುತ್ತಾ ಈ ಸೆಷನ್ ಅನ್ನು ಸುಂದರ್ ಸರ್ಗೆ ಹ್ಯಾಂಡ್ ಓವರ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಸರ್ ಪ್ಲೀಸ್ ಕಂಟಿನ್ಯೂ ಸರ್.
ಅವರು ಕಾಣಿಸುತ್ತೆ ನಿಮ್ಗೆ ಬೇಕು
ಸರ್, ಮುಂದುವರಿಸಿ ಸರ್.
ಮಾಡ್ಬೋದಾ?
ಎಸ್ ಸರ್, ಎಸ್ ಸರ್. ಸರ್ ಮುಂದುವರಿಸಿ ಸರ್, ಸ್ಟಾರ್ಟ್ ಮಾಡಿ.
Yesterday, I had given an introduction to the theories of evolution. There are so many theories. One was Lamarckism, then it was Weismannism, then Darwinism, and then Hugo-de-Vries mutation theory. None of these theories were satisfactory, not convincing. Lamarck could not succeed because he had very wrong information about evolution. Weismann also was not giving the correct information. Darwin looked like very convincing, but there were some drawbacks, some defects in his theory also. Darwin said that evolution is a very slow process. It is a continuous process and a regular process, taking thousands of years for the development of a new species. Then, mutation theory of Hugo-de-Vries, it was completely different, altogether different from that of Darwin. Mutation theory said evolution is a sudden process. It is not slow, like as Darwin had said, but it is a sudden, quick process. And it is not continuous, extended over a period of thousands of years, but suddenly it occurs and it is discontinuous. So none of the theories were satisfactory. Later, some of the scientists found that if Darwin's theory of natural selection and mutation theory of de Vries and the hereditary laws of heredity of Gregor Mendel, if all these three things are combined together, it is possible to explain the mechanism of evolution. So they called it by a new name, they called it as Neo-Darwinism. Now, Neo-Darwinism gives a very clear case of evolution. Now we shall proceed further and find out how Neo-Darwinism is able to help us in understanding the mechanism of evolution. Now, each has its own drawback, as I told you. Darwin considered natural selection as the supreme force and ultimate force in evolution. It was natural selection which initiated the process of evolution, and it was natural selection again which produced the final product. But the natural selection cannot operate in the absence of variations. If all offspring are similar, how can natural selection pick up the fittest? The mutation theory is also not flawless. Mutations alone cannot bring about evolution. Mutations no doubt produce discontinuous variations, but in the absence of natural selection, mutations will be powerless. Thus, both theories fell short of satisfactory explanation of evolution. Now, Neo-Darwinism gives a different view. Natural selection in action. There are two examples to explain the operation of natural selection. They are, number one, industrial melanism, and number two, Ddt resistance in mosquitoes. The first one, industrial melanism. There is a type of moth called the peppered moth, zoologically called Biston betullaria. It is creamy white with black dots and dark regions on its body. This moth is active during the night and is attracted to light. During the day, it rests on the tree trunks. Its body coloration and texture blend so perfectly with the lichens on the tree barks, and it is almost impossible to spot them. Birds are their natural predators. During Industrial Revolution in England, many textile industries sprang up. Burning of coal as a fuel liberated large amounts of sulfur dioxide, and air was heavily polluted. As a result, the lichens growing on the trees around the industrial area were destroyed. The dark tree barks were exposed. The barks were coated with soot, making them darker. The landing places of these moths changed their color. It was a real disaster for these moths. When the moths rested on the trees, they were exposed and against the dark background of the bark. They were easily picked up and consumed by the birds, and their numbers started dwindling to a point of extinction within a short period. In the year 1895, a black moth appeared for the first time at Manchester. When it rested on the dark barks of the trees in the industrial region, it was perfectly camouflaged on the tree trunks which had been darkened by the soot. The number of dark moths slowly and steadily increased. By 1950, almost all the peppered moths were dark ones. Mutation occurred in the gene responsible for the pigmentation of the moth wings. To begin with, all moths were light color, that is, there was no variety or variation. They were protected by the environment. A change occurred in the environment. The tree barks were darkened, making the moths vulnerable to predators. A mutation occurred to produce dark colored moths. The moth population had now two varieties, dark and light. Thus, there were variations. Both were exposed to the dark background. Natural selection operated on the population. The dark moths were protected and the light ones were slowly reduced. The dark moths, being more in number, survived and contributed more offspring to the next generation. Now here is the figure of the moth. This is the original white colored moth, and by the side, the dark moth. Both of them are resting on the dark bark of the tree. So in this one, the dark moth is well protected, camouflaged, while the other one is exposed, it can easily be spotted by the birds and they are hunted and destroyed. Thus, mutation occurred first to produce variations, only then natural selection operated to select the fit ones. Now, the second example given is Ddt resistance in mosquitoes. During World War II, more American soldiers died of malaria than due to bullets. The epidemic of malaria was widespread in army camps and there was no drugs to cure it. The cause of malaria is the protozoan parasite Plasmodium. The female Anopheles mosquito acts as the vector of this parasite. Plasmodium needs two hosts to complete its life cycle. Man is the vertebrate host and the female Anopheles mosquito is the invertebrate host. To control malaria, either mosquito must be destroyed or man must be destroyed so that the life cycle of the Plasmodium cannot complete or will not be complete. So the DDT was a very effective insecticide that could annihilate the mosquitoes easily. When DDT was sprayed for the first time, almost all mosquitoes died. Nearly 90 to 95% of the mosquitoes died because of the DDT effect. Only a very few, a small number of about 5%, they resisted the deleterious effect of the DDT. The small percent of resistant mosquitoes that survived produced the next generation. Now again, malaria started. Once again, malaria became rampant. So DDT was sprayed again to control these mosquitoes. But this time, the number of resistant mosquitoes had increased. Only about 80% of the mosquitoes died because of DDT, and nearly 20% of the mosquitoes had become resistant. It was found that DDT was not at all an effective insecticide against mosquitoes. Generation after generation, the number of resistant mosquitoes increased. To begin with, mosquitoes were non-resistant to DDT. When DDT was sprayed, a mutation occurred in some genes non-resistant to DDT. Thus, the mutation produced variation in the population, namely resistant and non-resistant varieties. Both varieties are exposed to DDT. The resistant varieties survived and the non-resistant succumbed. The resistant members produced offspring to the next generation. Thus, generation after generation, the resistant mosquitoes increased their numbers. Now, you recollect the story of Nala and Damayanti. In your younger days, you should have heard of this story of Nala Damayanti. Nala was a very handsome prince, and Damayanti lived in the adjacent kingdom. She was a very beautiful princess. And these two persons, Nala and Damayanti, they fell in love with one another. Damayanti was so beautiful that even gods, the Ashtadikpalakas like Indra, Agni, Yama, Varuna, Vayu, etc., they also wanted Damayanti to marry any one of them. The Swayamvar was arranged and the various princes from different kingdoms arrived and they were sitting in different seats in the court hall. Damayanti also arrived. Damayanti had come there with a garland in her hands in search of Nala so that she could garland him and select him as her husband. So she went on going from place to place to find out where Nala is. Then she suddenly found Nala. She went near, and as soon as she came near, Nala, she found another Nala sitting by his side. And another Nala, like this, about 7 or 8 Nalas were sitting side by side continuously. All the gods are disguised as Nalas and they were sitting on either side of Nala. So Damayanti, that is nature, failed to select because there were no differences or variations in these Nalas. All of them looked alike. So genetics also, population evolution also had the same problem. Now we shall go to population genetics and evolution, in which Hardy-Weinberg law is a very important law. All these years, evolutionists try to explain evolution by taking pairs of characters or individuals to study how organisms evolve into new ones. Such studies did not lead the scientists to understand the mechanism of evolution. The present trend is to explain the mechanism of evolution in terms of the population as a whole, not individuals. Today, we talk in terms of population genetics and evolution. Population genetics deals with gene frequencies, how frequently a particular gene appears in a population. In the year 1908, British mathematician G.H. Hardy and German physician W. Weinberg independently found that the frequencies of alleles in sexually reproducing organisms remain constant for many generations under certain conditions. They proposed a law which is now known as the Hardy-Weinberg Law. This law explains the mechanism of evolution satisfactorily. Hardy-Weinberg Law states, in a large, isolated, pan-mictic population, if the frequencies of a pair of alleles are found in a definite proportion, they continue to remain in the same proportion generation after generation, provided there are no upsetting factors such as mutations, selection, or genetic drift acting on the gene pool of the population. I repeat the law. In a large, isolated, pan-mictic population, if the frequencies of a pair of alleles are found in a definite proportion, they continue to remain in the same proportion generation after generation, provided there are no upsetting factors such as mutations, selection, or genetic drift acting on the gene pool of the population. Now here, some important words have to be explained. The law says large pan-mictic population, large isolated. What is meant by a large population? A large population includes millions and millions of individuals. It is not about thousands, 10,000, 50,000, or 1 lakh, or 5 lakhs, or 10 lakhs, but crores of individuals in a population. That is known as a large population. Then the population should be isolated. That means there should not be another similar population in the near vicinity. They should be far away, completely isolated. And then pan-mictic. A population is said to be pan-mictic if there is random mating. That is, any member of the population can mate with any other member without preferences or selection. Then gene pool. The sum total of all the genes contributed by all the members of a population to the next generation forms the gene pool of the population. The three forces mentioned in the law, namely mutation, selection, and genetic drift, these are known as elementary forces of evolution. As long as these forces do not act on the gene pool of a population, the frequencies of the genes are not affected and there will be no change. The population is static and is said to be in equilibrium. Such a situation is known as Hardy-Weinberg Equilibrium or genetic equilibrium. As long as the population is in equilibrium, it is not evolving. If one or more of these elementary forces act on the gene pool, the frequencies of the gene change. Then the equilibrium is upset. It means that the population has started evolving. The three elementary forces of evolution, namely mutation, selection, and genetic drift, however, by themselves are not strong enough to bring about the origin of a new species. They may bring fluctuations, changes in some of the organs, some of the structures of the body, but a new species cannot develop only with the help of these three populations, with these three factors. Additional forces like isolation and migration are required for the emergence of a new species. So you can remember that there are 5 important factors which are necessary for the formation of a new species. So it can be easily remembered by this. Mahatma Gandhi made India secure. So Mahatma M represents mutation, Gandhi G represents genetic drift, again M represents migration, I represent isolation, and S represents selection. So if you want to remember the 5 important factors or factors deviating from Hardy-Weinberg equilibrium, remember Mahatma Gandhi made India secure. M represents mutation, G represents gene drift, genetic drift, M represents migration, I represents isolation, and M represents selection. Sources of variation as evolutionary force. So, if you want variations, there are some important factors. The sources of variations include basically 2. One is mutation, the second one is recombination. Mutation can be of 2 types. It may be gene mutation or chromosomal mutation. Recombination of genes also occurs because of 2 factors. One is by heterozygosis, that is, crossing of 2 different types of homozygous individuals, resulting in heterozygous individuals. And when these heterozygous individuals mate, you get varieties or variations. Then the second type of recombination is by crossing over of chromosome segments during meiosis. This also results in variations. Then chromosomal mutations again may be classified into 2 types. One, change in the chromosome numbers. And the second one is change in the chromosome structure. Chromosome number can be changed, and accordingly 3 different types have been found out. Chromosome number can change in one of them known as non-disjunction. Non-disjunction means during cell division, either mitosis or meiosis, the chromosomes arrive at the equatorial plane. They arrange themselves along the equatorial plane, and later they are pulled apart to different poles, to opposite poles. One set of chromosomes move towards the animal pole, another set goes to the vegetal pole. So in case of a human being, there are 23 pairs of chromosomes. Out of them, 23 chromosomes move to animal pole, the remaining 23 chromosomes move to the vegetal pole. But occasionally, accidents do happen. You know, the chromosomes are pulled to the respective poles by means of spindle fibers. Sometimes what happens is one of the spindle fibers may be cut, snapped. So when the spindle fiber is cut, the chromosome cannot move to a particular pole. So it will be unnecessarily moving towards one pole. Instead of the two chromosomes, homologous chromosomes moving apart, both of them move to the same side. So ultimately, the resulting gametes will not be alike. Instead of carrying 23 chromosomes, one of them carries 24, the other one carries only 22. So there is a difference of one chromosome, addition of one chromosome or loss of one chromosome. So this is because of non-disjunction. Separation of chromosomes, separation of homologous chromosomes is known as disjunction. When they fail to move apart, that is known as non-disjunction. Then the second one is haploidy. Chromosome number again changes. One complete set of chromosomes disappear. So instead of two sets of chromosomes, some organisms may have only one set of chromosomes. This condition is known as haploidy. And the third is polyploid. That is, if more than two sets of chromosomes are there. If there are two sets, we call it as diploid. If there is one more, three sets, it becomes triploid. Like this, polyploid, a number of sets of chromosomes may be present. So this also results in variations. Then this other type is change in the chromosome structure. Chromosome number is the same, everything is the same, but the chromosome structure may change. For example, there may be duplication, there may be deletion, there may be inversion, or there may be translocation. This we will come back again later. So, genetic drift is also known as non-adaptive evolution. Genetic drift forms the third elemental force of evolution. It is evolution that occurs through random changes in allele frequencies over time. It was first identified by the American geneticist Sewall Wright. Hence, it is also called Sewall Wright effect. The members of a population do not pass on the allele in the same proportions. Sometimes one allele is passed on in large numbers, while some other time the other allele is passed on in large numbers. As a result, the allele frequency fluctuates. This fluctuation in allele frequency is known as genetic drift. Why this genetic drift occurs? Genetic drift is a matter of probabilities. During gamete formation, the segregation of any chromosomes into eggs and sperms occur by chance. During mating, a large number of gametes are wasted. Only a few succeed in combining to form new individuals. Now let us find an example. Now you put about 50 black balls and 50 white balls in a bag and shuffle them. 50 white balls like the table tennis balls, which are white in color, and then similarly 50 black balls, that is table tennis balls which are colored black. You put them in a bag and shuffle them. Now the bag contains 100 balls. Now, without looking into the bag, you insert your hand, pick up one ball at a time. The first ball, let us suppose, is white one. What are the chances of the second pick resulting in a black ball? After 10 picks, you count the black balls and white balls you have picked. You find that there may be all the 10 may be black, no whites, or it may be 9 black and 1 white, or it may be 8 black balls and 2 white balls, or 7 blacks and 2 whites, 6 and 4, 5 each, 4 black, 6 white, 3 black, 7 white, 2 black, 8 white, 1 black, 9 white, 0 black, all 10 white. When you have picked all the 100 balls, then the ratio between the black and the white ball is 1:1, 50 each. See, that is the beauty of genetic drift, that is probability. Organisms during reproduction do not necessarily release their alleles in the exact proportion in which they possess them. The gametes carry any one of the 2 alleles for a trait. There is a chance mating. Which gamete goes into zygote formation is purely a matter of chance. In human population, the chances of a couple getting either a son or a daughter are equal, 1:1 ratio. But we have seen families having only boys. Similarly, there are families that possess only girls. In some generations, there are more boys. In some generations, there are more girls. But when you take up the entire population of the world, they will be approximately in the ratio of 1:1. Now, gene mutation. See, gene mutation is a type of mutation in which a particular gene undergoes a structural change. Now, here is an example. Imagine this is a chromosome. This chromosome has a number of genes. Each letter represents a gene. I sleep on the mat. I is a gene, S is a gene, L, E, E, P, O, N, T, H, E, M, A, T. All are genes. During cell division, it may so happen that the gene M may be rubbed off, and instead the structure may change. And instead of M, it is replaced by another gene, that is R. So when this occurs, instead of this statement, I sleep on the mat, the chromosome now reads, I sleep on the rat. So a single mutation, single gene mutation will change the entire meaning. Now coming to chromosomal mutations, similar thing happens. Chromosomal mutations may be of different types. One is duplication. A segment of the chromosome is replicated and it attaches to one end of its homologue. See, this is the chromosome, and this chromosome has a number of genes on it. It reads, sign as soon as you report to duty. These are the genes. Now, during cell division, what happens is a particular segment. For example, the segment RE detaches itself from one homologous chromosome, moves and attaches to the other homologue. So the other homologous chromosome will now read, instead of sign as soon as you report to duty, it now reads resign as soon as you report to duty. So this duplication of these two genes or gene changes the entire meaning of the sentence. Similarly, The next one is deletion. A segment of a chromosome detaches and disappears. Here is a sentence, or here is the chromosome on which the genes are present. So it reads, I purchased a car to go to office. Now here, a small segment of the chromosome, the first portion, PUR, it gets detached and disappears. So the segment detaches and it disappears. Now the resulting chromosome reads like this. I chased a car to go to office. Instead of purchased, it now reads I chased. So loss of a small segment of a chromosome also gives variations. Now inversion. During this process, a segment of a chromosome snaps, then it rotates by 180 degrees, but rejoins the same chromosome, changing the sequence of the genes. Here, twinkle, twinkle, little star. This is the standard chromosome. Now in this one, the last segment, STAR, gets detached, then rotates by 180 degrees and rejoins. Instead of STAR, it now reads RATS. Now the revised chromosome arrangement is twinkle, twinkle, little rats. The genes are the same, no change, but the chromosome arrangement is different. Then the next one is a translocation. A segment of a chromosome gets detached, comes and attaches to a non-homologous chromosome, making it longer. Translocation. Here, lion eats flesh. It is a carnivore. This is a chromosome. But there is a non-homologous chromosome in which a part of it detaches and gets attached, and it reads, the cow is a herbivore and it eats grass. So it changes. So the effect of genetic drift is considered as a fickle force. It results in loss of variation, genetic variation. Smaller the population, the quicker is the loss of genetic variation. Because of genetic drift, the individuals become genetically similar. Natural selection has no scope to operate. Because of the peculiar way in which genetic drift acts, it is also known as non-adaptive evolution. There are populations that were historically large, go through a period of time when they are small and then become large again. Such events, when the populations become temporarily small, are called population bottlenecks. During the lean period, drift operates to minimize the genetic variation. This is called bottleneck effect. See here the figure gives. This first figure shows a bottle in which black and white marbles are introduced. There are 36 black marbles and 24 white marbles. Original population frequencies of black and white alleles is in the ratio of 36:24. Now you tilt the bottle to remove some of these marbles. So when you tilt the bottle, a few marbles come out. A chance environmental event greatly reduces the population size. So some of the marbles come out. Now, after that, you once again find out how many marbles are there in the bottle. surviving population has different allele frequencies. Now, the frequencies of these marbles have changed. Instead of 36 and 24, the black marbles are only 4 in number, while white marbles are 9 in number. Though originally it was 36 and 24, because of this accidental tilting or falling of the bottle, the number has changed, the frequencies have undergone changes. Surviving population has different allele frequencies from the original population, which generates a new population. Now, another important point you should remember about genetic drift is, in some generations, it produces variations which are very favorable, very useful. But in some other generation, it may produce variations which are very dangerous. So it is not actually adaptive. That's why it is called non-adaptive. To give you another example, according to Darwin, survival of the fittest organisms will survive whenever there is a struggle for existence. Now, this is not true in all cases. For example, imagine you take the army of a country, of our own country. The army is made up of youth, young men between the age of 18 and 30, very active. Most of them are not married yet. They are physically 100% fit, mentally alert, very active, and they are 100% fit individuals. They are considered the fittest. But suppose a war is declared. During the war, many of these fittest persons are killed by the enemy. So during war, many of the fittest persons are killed by the enemies. That means there is no survival of the fittest. Even though the fittest should survive, here is an example where the unfit will survive, the fittest are destroyed. At the same time, the unfit ones, persons who are unable to walk, those who are unable to move, those who are unable to do any other physical work, they may survive. Some of them, who might have lost their limbs, may still remain in home. They may produce children, they may contribute offspring to the generation, but the real ones, the ones which are considered to be fittest, without contributing their genes to the next generation, they will have died. So this is how this genetic drift works. There is a small island. The island consists of about 100 individuals. Out of these 100 individuals, there are 30 individuals who are suffering from a particular disease. About 22%, these 30 members, they are homozygous for that particular disease. Then the remaining 22%, they are heterozygous for the disease. That is, they are carriers, but they do not suffer from the disease. And the remaining 18, they are the healthy individuals. So, in a population of about 100 individuals in an island, 30 are having diseases, very fatal disease. 22 are carriers, but they are not sufferers. Then about 18 are very healthy individuals, but they are quite normal. So this is the analysis. Suppose all of these 100 individuals have come to the seashore for some festival. All these members have come to the seashore for a festival, they're enjoying. And during this process, suddenly there is a tidal wave dashing at the island. In this, about 50 individuals are killed, only 50 individuals survive. And when you analyze them, you find that about 22 of the survivors are healthy individuals, about 6 of them are carriers, and the remaining are the ones suffering from disease. So the entire proportion is changed. Suddenly, even the unfit may become extinguished, or the fittest may reduce in number, unfit may increase in number. This is purely because of some accidental occurrences. So it may happen in any population. So population need not always expect that the fittest will survive. The unfit also can survive. The fittest may be eliminated. So this is the peculiarity of this genetic drift. Now, similarly, we come to the sexual reproduction in organisms. Organisms which can reproduce sexually, they have a greater role in evolution than those organisms which reproduce only by asexual methods. Let us see how it is possible. In sexually reproducing animals, genetic recombination is very important in addition to mutation to provide variation. So if you want variation in sexually reproducing organisms or in the population of sexually reproducing organisms, then in addition to mutation, there is need for a recombination of the genes. Genes of different persons should come together. Then only variations can be produced. Now, recombination is possible during sexual reproduction. Thus, sexuality plays a very vital role in the evolutionary change in animals. Imagine a population of animals in which all the members are homozygous for two important genes, capital P, P and capital Q, Q. So all the members of the population have this particular gene, capital P, P, Q, Q. This homozygous condition confers maximum adaptation to the environment. So these animals are very happy, they are quite prosperous in that particular environment because they are homozygous for these two pairs of genes. Now, gene capital P is responsible for a large body size. Similarly, the gene capital Q is responsible for stout legs. The legs should be stout enough to carry the large body. So it should be very important. Now, environment undergoes a change. As a result, food and water become scarce. Now, these animals have to change according to the environment. They should reduce their body size and the legs should become slender. Instead of the genotype capital P, capital P, capital Q, these animals should change to the new genotype small p, small q, small q. Small p is a recessive gene which confers a small or reduced body size, and small q is a recessive gene which confers slender legs. If all members reproduce sexually, then can mutation and selection provide the required change? Mutations are very rare. One in 100,000 genes undergoes mutations. Let us say that one animal mutates for the gene capital P and its genotype becomes capital P, small p, capital Q, capital Q. Another animal undergoes another mutation from capital Q to small q, and it has now a genotype capital P, capital P, capital Q, and small q. What are the chances of both mutations occurring in the same animal? It is 1 by 100,000 square, that is 1 in 10 billion. The two mutations may never occur together. To get the genotype small p, small p, small q appears almost impossible in asexually reproducing organisms. Suppose these members reproduce sexually, the results are quite different. The two mutations may occur in two different individuals. The mutants have the genotypes. One individual has capital P, small p, capital Q, and the other one will have capital P, capital Q, small q. If these two individuals mate, 25% of their offspring will have the genotype capital P, small p, capital Q, small q. Heterozygous for big body, heterozygous for big or stout limbs. When these double heterozygotes mate, offspring with desirable genotype small p, small q, that is, smaller body size and slender legs emerge in the next generation. Thus, sexual reproduction and genetic recombination provide the desired change much quicker, that is, only in 2 generations, than mutation and selection. Now, this chart gives you a clear idea. See, evolutionary advantage of sexual reproduction. Original animal is capital P, capital Q. In one animal, capital P mutates to small p. In another animal, capital Q mutates to small q. You see, asexually reproducing animal. Capital P has reduced to small p. Its genotype is capital P, small p, capital Q, or in all offspring. Again, the same thing happens. Now, similar thing happens again. Now, again, what happens is the other organism where the crossing over has occurred and a mutation has occurred in small q, the organism will have the genotype capital P, Q, small q. So this may go on for any number of times. This organism, by repeated asexual reproductions, gives rise only to capital P, small p, Q genotype. Even after 100 generations, it will remain the same unless another mutation occurs. Similarly, the other individual, where capital P, capital P, capital Q, small q, by asexual reproduction goes on producing only this particular type of genotype without any change. So absolutely there is no chance for any organism in the population to get smaller body and slender lips. But if you take sexually reproducing animals, you see here, capital P has mutated to small p, so it will be, the gametes produced will be capital P, capital Q, other gamete will be small p, capital Q. Similarly, other organisms, small p, capital Q, and small p, small q. A big p means small q. So if there is mating of this, you get capital P, small p, capital Q, small q. And if two such animals mate, you get 16 varieties having different dihybrid ratio. If you go through this, you will find that at the end, there is, out of 16, one individual with the genotype small p, small p, small q, small q. That is the desired genotype, that is animal with reduced or small body and animal with slender limbs. This can perfectly adapt itself to the new environment, changed environment. Thus, sexual reproduction gives a definite benefit to evolution. So evolution can be very effective only in sexually reproducing organisms because there are plenty of scope for variations. It may be mutation, gene mutation, chromosomal mutation, recombination, crossing over, heterozygosis. Like this, there are so many options in sexually reproducing organisms to provide variations. Thus, today, the emphasis has been laid on the population, not on individuals. So, the unit of evolution is the population and not individuals as it was earlier thought to be. So, this is a very important thing. So, these are the important points about organic evolution in which we deal with population genetics and Neo-Darwinism. So with this, we complete the lecture for the day. Thank you.
Hello.
Hello.
Yes, sir. I am here, sir.
ಇವತ್ತಿದ್ದು ಮುಗೀತು.
One second, sir. Umesh sir is coming, sir.
Yes. Very good evening, sir. Thank you very much for your wonderful session on evolution part 2, sir.
Okay, thank you.
Advanced lecture series. There are any questions from participants, students or teachers? They can mention in the chat box or they can raise their virtual hand so that we can unmute and they can speak directly to the speaker. If there are no questions, please type yes or no in the chat box so that we can wind up the session and we can go to the final session of this section. Valedictory. Sir, as of now, there are no questions. Students are hesitant to ask the questions in the Webex Cisco platform. And in the YouTube also there is, we have not provided any provision to ask them questions. So they may contact us offline, either through mail or through call or through WhatsApp or something like that. If that is the case, we'll contact you back and we'll get the answers and revert them back.
And further, one more point.
Yes, sir.
I have not completed the evolution. Only Neo-Darwinism I have completed. Is population genetics I have completed.
Yes, sir.
Still, there are some other topics. For example,
Yes, sir.
adaptive radiation, convergent evolution, divergent evolution, and evidences in favor of evolution. All these things are there.
Okay, okay.
Some other time we can go through that.
Yes, sir. Yes, sir. Another 2 to 3 sessions you may require, sir.
One more session.
One more session or 2 to 3 more sessions, sir?
I think so. One is enough, I think.
Okay, sir. Fine, sir. So one more session we can complete this evolution series, sir.
Okay.
So coming to this valedictory, on behalf of KSTA and on behalf of all the participants and on behalf of teachers and students who have joined with us in the WebEx as well as YouTube channel, I wholeheartedly thank you very much, sir, for accepting our invitation to deliver this session on evolution. Thank you very much, sir.
Thank you. Thank you.
And we wish to collaborate with you in future sessions also, sir, related
Sure.
to this subject.
Okay.
Also, I would like to thank our chairman, Professor S. Ayappan, sir, our CEO, Dr. A.M. Ramesh, sir, our senior scientific officer, Dr. Anand, and our colleagues, Dr. Srinivasu, and the technical persons, Anthony Lobo, Shri Siddhegowda, and from KSTEPS, Mr. Sumarani, and our non-technical persons who have helped in this webinar series, lecture series. So, and last but not least, I would like to thank all the participants, teachers as well as students who have attended and joined us in this Cisco WebEx platform and YouTube channel platform. Thank you one and all. Hope to see you all tomorrow session. Thank you, sir. Thank you. Have a good day, sir. Take care. Thank you.